I'm a clinical geneticist, and I'm from a very small country, but nevertheless I'm confronted with big data. It all started about 15 years ago when we started to print our own array CGH slides to do array comparative genome hybridization, and we were collecting many thousand, 10,000, 100,000 data points on one single patient, and we had somehow to make use of it, and we needed to know what kind of variants were specific to the population that we were serving and were not pathogenic. We started to set up our own database, and with the help of a couple of software engineers, we developed a new program that was then commercialized as CARTaGENE, not CARTaGENE, but CARTaGENE. And we have now about 10,000 samples in it. I don't have any shares in CARTaGENE, so. And we also upload our unknown cases with genomic copy number variants in the Decipher database in the UK, which is very helpful because then different clinicians can contact each other if they find someone who has entered a patient with a deletion or a duplication in the same genomic area. In 2013 in Belgium, all 8 genetic centers started to use array CGH for their prenatal karyotyping. So we don't look at chromosomes anymore, we all look at array CGH data, which poses some questions and some problems. So we set up a working group. They all use the same software, and this working group will harmonize the variants or the deletions and duplications that are reported back to the clinicians and to the patients. We also use this same working group to pool our data on non-invasive prenatal testing. We have now done more than 5,000 analyses of serum samples from pregnant women. We sequence them, we have 10 million reads of about 50 base pairs, so that's about one-tenth of the genome. If you then have 5,000 samples, you can start mining these data to look at variants in the population that we serve that are quite common. But then with next generation sequencing, we have, of course, much more data available from exomes, from gene panels, and so on. And we developed also a system to share some of these data without having to move the data to one central place or having to get consent to centralize clinical data sets, and this is done with the program NGS Logistics, which allow for decentral storing of the data, but with a centralized way of querying the data, especially interesting for clinical purposes, because you can query the data set for one specific variant and know if it has been seen or not before, or for variants in one specific gene, but it does not report clinical data. So you have to contact a specific center to know what the clinical data in the specific patient was. And then the next thing we want to do is to enter all data from exome sequence analysis into our electronic medical database from the hospital to see if these data can be queried by the clinician, or for instance for pharmacogenomic purposes, if someone prescribes electronically a certain drug, then the machine can look for specific variants in the genome that have been sequenced and alert the clinician. But this is still a dream. For those of you that might show some interest in NGS Logistics, it's a schematic of how it works, but I'm not a software engineer. For me, this is too complicated.
Thank you, Éric. The first questions building on that are going to be talking about the integration of administrative and medical data. And so I'll direct the first questions to Éric and to Nancy. Drawing from your experience in the European and Canadian contexts, what are the major roadblocks to integrating genetic, clinical, and administrative health data, and how did you overcome these in your cohorts?
I'll start. The short answer is we haven't overcome them, but I think just in kind of thinking back through the presentations already we've seen this morning, that there's sort of a call for research in this area that's looking at integration and tapping into the potential of the data, is that we need to be able to pool various genomic data, whether it's across projects, across provincial boundaries, or even international boundaries. And seeing the value in linking to the phenotypic data that is maybe clinical data, it might be registries, it might be EMRs. And that involves data linkage and integration. And then thirdly, Michael touched on it a little bit this morning, but the value in linking to where we can to population-based data so that we can understand a given bias in a study. So those three things together, it's a pretty complex problem with many different heterogeneous entities that we have, at least in the Canadian context, which in the world of data translates to many different decision makers around data. And so that might be PIs on given projects, it might be data stewards in a public body, or even individual physicians, which gets to be a very challenging situation in British Columbia anyway. But if we're thinking about what are the roadblocks to integrating, and I think the ultimate goal we have to keep in mind is what's the point of integrating? It's to be able to access it, to be able to use it, and importantly, to be able to access in a timely and reliable fashion. And in my view, there are really two key elements to address. And I think they're best addressed centrally, but we can get into some dialogue around that. And it has to do with data access governance. So one is the privacy-related issue, and the other is social acceptability. And I'm just going to, if I may, delve in a little bit on the privacy one in particular. And I think, again, it's useful, it ties back to a few of the presentations earlier, is that there's really a significant cultural barrier or challenge with respect to what we have ahead of us. And that working with the genomic culture of data, it's been very much one of sharing, of openness, and it's backed by consent, as Michael referenced earlier. I think that arguably the social acceptability of the openness, of the full openness, is changing, that many of these are moving towards sort of access restrictions. But nonetheless, it comes from a very different sort of provenance. And you add in administrative data, which is the world that I've lived in for many years, which is a very traditional world and cautious, and where generally consent is not considered practicable, so it is not present. And when those two worlds come together, I characterize it as privacy shock. And I think that if we unpack that a little bit, looking at the foundation principles of privacy, one is they lead back to the OECD guidelines around privacy. And it brings in ideas around, it all relates to personal information, brings in ideas around limiting collection, specifying purposes, minimum use of minimum amount of data needed for the research question. And so I think that I think you can see quickly how the genomics and the administrative principles on which research data access are coming into direct conflict. So it's not entirely clear to decision makers in this space about how one should act responsibly, ethically, or even legally in this context. I think that's going to be a significant challenge.
Éric, did you want to add anything to that?
Yeah, the program that I'm showing here, the NGS Logistics, was especially designed to get around some of these issues, because you don't upload your data to a central server. You keep them in your own place. You just make them accessible. Only part of it is accessible. So these are the raw data files, the FASTQ and BAM files that are available to query, not the variant call files, because they have been interpreted yet. They have been passed through pipelines that change every year, so they're getting better and better. So you use the raw data files that are decentrally stored and are not supplemented with clinical data. So the clinical data stay in the individual centers. If you want to know if a variant has been seen or how frequent it is, one specific variant or variants in a specific gene, you can do a query, get all the data without breaching confidentiality or privacy issue. That was the idea behind this program. And for this specific purpose, it seems to work fine. Of course, you cannot do anything you would like with all these data, but that's another issue.
Thank you. Before I move on to the next questions, I just want to open it up to the other panelists and see if anyone wants to weigh in on what's been said so far. Okay. So the next question, again, for Nancy and Éric, can you give us some concrete examples of how the integration of health record data or clinical data has enabled a better understanding of the etiology of disease and how this is translated to a more precise or better delivery of point-of-care service, and how that can be done rapidly? And obviously, we've heard some examples this morning, if you could build on that.
In clinical genetics, it's very important to know what is pathogenic and what's not pathogenic, and what is, and if it's not really pathogenic, if it might be a risk factor or not. And so we have set up this working group on prenatal testing in array CGH to make sure that all the different centers in Belgium use the same criteria, contact each other in the working group if they are not sure how to report a certain copy number variant or if they should report it or not. Mm-hmm. And so putting all these data together, we have a very good idea what we have to do in the prenatal context, because there it is very, very important not to make any errors and not to report in a different way in different centers in the same country. Yeah. So that's very important. Yeah.
Nancy, do you have anything you want to
I think others on the panel are much better positioned.
Okay. Would anyone like to build on that?
Well, I'd just like to maybe build on some of what was said with respect to data sharing. I think that Nancy is right. It's a very complicated space, and it's also a very dynamic space in that some attitudes about privacy are changing really rapidly with, with the widespread use of social media, which is still a relatively recent phenomenon. There are many activities that are meant to try to, and organizations that are really trying to help solve these problems, and Canada is actually pretty prominent in some of them. And so CARTaGENE is involved in the Global Alliance for Genomics and Health, I think. And also the International Rare Disease Research Consortium is a group of funders that are placing expectations on the researchers that it funds to encourage them to share data, to get make sure that we get the maximum science and benefit out of the work that we're funding.
Thank you. Does anyone else want to weigh in before we move on?
And I'll just add to the sort of the examples of success stories. I think we think of big data as a concept of something where you bring in lots of data together and you turn some computer wizardry and magic happens. But my sort of most exciting sort of exposure to big data is kind of very much the opposite, which is I work in a multidisciplinary team in Oxford at a hospital where we look at patients who probably have some genetic cause of a syndrome, but for whom conventional testing has failed. And we decide whether to sequence their genome or not. And if we go ahead with that, then, of course, there's the whole process of sequencing and analyzing and interpreting it, annotating it against what is known out there in the rest of the world. But actually, the final interpretation, the decisions about what to conclude, and of course, how to then influence the patient's treatment, is very much dependent on a dialogue between all the parties involved, the bioinformaticians, the statisticians, the clinicians. And I think if we're ever going to make big data a success, and I think these are small cases of big data success, but if you're going to roll that out on a national scale, as we're trying to do in the UK in the 100,000 Genomes Project, then somehow we have to find ways of capturing that interaction and that potential for dialogue, because without that, you can have a lot of things falling through holes.
Thank you. So the next set of questions I'm going to ask are for Daniel Levy and Paul Lasko. So as directors of a cohort and of a funding institute, how do you maintain the level of funding that's required for these cohorts? I know everyone's very interested in your answer, Dan. And how do you promote the use of these programs by researchers as a vehicle to facilitate discovery? And I'm just going to give there kind of a couple of related questions that I want you to address. So what are the challenges from the US perspective? For example, how do you maintain relevance in Washington? How do you keep the public engaged? And how do these cohort programs fit in the overall health policy, or do they?
Yeah, very challenging questions. First of all, I'm delighted to be here, and truly by all rights, I have no right to be here. I probably have never used the words big data together in the same sentence in my entire career. Yet I find myself here as a panelist and tomorrow as a speaker to address issues related to big data. When Framingham first started 66 years ago, it was simply designed as an opportunity to identify factors that might relate to the development of cardiovascular disease. It was a prospective study at a time when most research on cardiovascular disease was purely cross-sectional. It applied methods of epidemiology that previously had been used for solving problems of infectious disease outbreaks, yellow fever, typhoid, malaria, cholera in central London, and John Snow solving the outbreak by removing the handle on a pump. Yet the principles of epidemiology, with a little bit of vision, were applied to the problem of a chronic disease, cardiovascular disease. And it was predicted when this little study in the town of Framingham, Massachusetts started in 1948, that it would take 20 years to accrue enough events to be able to link measurements made at baseline to the occurrence of disease over time. As I will tell you a little bit more tomorrow, some of the insights from that work resulted in identification of the earliest risk factors for disease. And 20 years after the study began, NIH decided to close down the Framingham Heart Study. It had fulfilled its expected 20-year mission. Many of the staff were laid off, a skeletal crew remained, and owing to an intense lobbying effort and very intense publication of very important papers in that period of time in the late 1960s, the study was able to continue, and in fact, it began recruiting a new generation of study participants, the children and spouses of children of the original participants. So the original participants in 1948, their children and spouses of those children started coming into the study beginning in 1971. And part of the answer to the question you asked is, why is Framingham still alive and how did it remain current? And the answer was really at that point in time by reinventing itself. It brought in a new influx of young people free of disease. It continued to follow the older participants, not for coronary disease, which was the original mission, but now was following them for development of stroke and dementia, diseases of an older age group for the most part. And it had that new influx of young people and was able to study them in relation to diseases within their families because they had data on the original participants and their children. Framingham was now reinvented with no geneticist, reinvented as a study of familial patterns of disease. In 2002, we began the recruitment of a third generation of participants, and with a little bit of vision, some of my colleagues began in the late 1980s to collect blood samples for isolation of DNA, and with then at that point in time 2 generations, eventually 3 generations of participants, that DNA served as a valuable resource for repositioning Framingham yet again in a new way to contribute in a vital effort to understand the genetic basis of primarily cardiovascular disease, but in reality almost any phenotype that had been studied in Framingham could be addressed through the methods that were applied. but we didn't have a single geneticist working in Framingham. We collaborated with some geneticists, but my colleagues and I viewed genotyping simply as a tool to be applied to the study of epidemiology rather than as a goal in and of itself. It was a tool for another goal, to understand the basis of disease by the insights provided through genetics. And in 2013, NIH once again cut funds for the Framingham Heart Study by about 40%. Our exams of our 3 generations of participants were terminated, and my colleagues at Boston University were forced to lay off 19 employees working on the Framingham Heart Study. How did we survive? Once again, we survived through a lot of hard work, through reinvention. Once again, we went into new fields of research, and we continue to do so to find new opportunities, whether it's efforts to incorporate gene expression, not only genetic variation, proteomics and metabolomics, to use these new omic high technology approaches, again, not as ends, but as means to an end, to help us understand the molecular basis of disease, applying principles of epidemiology with the advantage of these new 21st century tools. So now we have had funding restored to allow us to re-examine our participants yet again. Although we are experiencing a bit of belt tightening, we are able once more, through a little bit of belt tightening, through a little bit of clever approaches to science, through competitive grant approaches, through other agencies such as the American Heart Association, to be able to continue to explore new avenues of research. And as long as the research that we're doing remains vital, hopefully we'll continue to have enough funding to survive in these very difficult funding times. I hope I didn't make everyone depressed.
So Daniel, what proportion of funding for Framingham comes from NIH?
Yeah, I would say about 90 to 95% of our funding comes from NIH.
So I can give the Canadian perspective and the perspective from the evil funding agency.
Is it less depressing? Or more
we'll see. We'll judge the audience response. It's no secret that we are in challenging financial times, and it's also no secret that there are many competing demands on CIHR's resources. That being said, cohort-based research is a truly vital component of the overall CIHR research portfolio, and CIHR funds a lot of it. We fund several hundred different cohort studies. The highest profile one, of course, is the Canadian Longitudinal Study for Aging, and then it goes down to a lot of little ones that get funded through the Open Operating Grant Program. So cohort-based science is, and no doubt will continue to be for the foreseeable future, an important component of the overall portfolio science that CIHR funds. Even in these challenging times, we were able to come up with another tranche of funding for the CLSA, which was not an easy thing to do, but I think the fact that we did it underscores the commitment that CIHR has to sustaining that important cohort study. What we have talked about internally is we are concerned, of course we want to encourage data sharing because we would like to see as much science as possible to come out of the investment that we're making in cohorts. And data sharing can also help ensure the sustainability of cohorts and the data that they produce. And we're really quite concerned, especially with the little ones, that were more or less, run by one investigator and the data sits on one desktop computer somewhere, that data becomes lost upon the retirement or changing interests of that researcher. So we certainly have had internal conversations that have not yet led to a conclusion around the idea of expecting a certain level of data sharing from these from people who want new funding for cohort studies, both with the goal of advancing the science more efficiently, but also as a means of potentially ensuring the sustainability and putting this data on a more robust footing and perhaps contributing to it being sustained better.
Thank you. So finally, in the final round of targeted questions, I'm going to turn to Guy and to Gil. As big data users, what are the challenges that most affect your ability to conduct research, and what are your suggestions to overcome these challenges? And what are the advantages of accessing these data sets and the value to your current research?
Okay. So I'm a statistical geneticist, and as a statistician, there are basically only two things I care about. One is numbers, and the other is error. It's a pretty stark life, unfortunately. And as we all know, numbers at some level isn't a problem. There's an awful lot of relevant data being generated across the world, whether it's genomic or sort of the healthcare administration data that we were hearing about earlier. So data being generated is not a problem. What is a problem is the access side of it. And as postdocs in my group will tell you, many of whose lives I've completely ruined, the continuous process of applying for access, curating the data, normalizing it so that data taken from one place kind of means the same thing as data taken from another place, coping with the systematic error structures that you get. These are things that ruin people's lives as analysts. And I think one important comment is that a lot of, every cohort is in some way special and unique, but actually from an analytical point of view, what you really want to do is to have access to lots and lots of cohorts, because if you see the same thing in lots of different places, then you believe it. So the access side, I think, is a major, major headache, and our current model is unsustainable. We simply can't go on accessing in dribs and drabs as we are. I think there are some ideas. Clearly, the general research license that NIH is sort of putting in place is a very good one. In the UK, the sort of generic access that's going to be put in place around the 100,000 Genomes project is a step in the right direction, although I think it remains to be seen how that actually pans out. I think that will solve some elements of the problem, but it won't necessarily solve the heterogeneity issues. And I think that's where the discussions that one can have at an international level are absolutely critical. And so Global Alliance for Genomics and Health is one sort of useful and I think informative, one good place to look to try and make that harmonization work. I'm involved on that and the data side, trying to get the genetic data being able to be shared effectively and sort of simply so that it means the same thing in different places. But that's clearly just the start of an incredibly long process to get to the same level with the phenotypic data and all the healthcare administration data that we've heard about still. So, as I say, data isn't an issue, but the access and the harmonization, I think, are the major challenges for use.
Okay. I would agree with that. I'm an imposter here. I'm a neurologist. I don't know what big data is, and the whole notion scares me. And it talks about what are my problems, so I thought I would talk about my problems. So data size, when we started, big data for us is genomic information because that's the big data that sequencing information. When we started, we were trying to download exomes, and when we tried to do that, we crashed the hospital informatics system. Because we had put in what would take up 24 hours of 100% of the capacity of the hospital network. So we were banned and we were no longer allowed to do this. And so we went high tech, and my student took his bicycle and a pack sack and hard drives, and this is how we started with the transfer of big data. So this is how I learned that transfer is a problem, and when I was involved at one hospital, one of my highest priorities was to install a risk line, and a risk line is a Canadian connection for letting lots of information flow very quickly. And so there are solutions. And I was horrified to learn that, in fact, we were the first hospital to have a risk line. And I think the Institute of Cardiology has a risk line. I don't think the new CHUM has a risk line, and I don't think the MUHC has a risk line. So transfer of information, I think, will remain to be a problem. Storage. We have a partner called Compute Canada, and the local node is Compute Calcul Québec, which are fantastic partners from the point of view of helping us with the hardware. However, these fields were dominated by physics and by weather, and so they had fantastic data processing, but they had really lousy data storage. And so it ended up that just my lab, the amount of data that we had to store was superior to the data of the entire health network of the province, including all imaging in the province of Quebec. And that was just my lab. And so there are solutions to that, but I don't think those are that good yet. I think that there's something that's evolving. But I think that if you want to deal with big data, you got to deal with moving it and you got to deal with storing it. And I think those are things that are big. Computational power is really not a problem, has at least not been for me, because of these Calcul Québec. Next is data analysis or bioinformatic tools. I don't know how many bioinformatic tools are out there for dealing with genetic data. Probably thousands, I would guess. And it's very hard to know which ones work, which ones don't. What's for sure is none of them work well, and that they all do different things. And so we did this little thing, I think Marie-Pierre might have been involved, looking at genome data, and we used 3 different programs for looking at CNVs, and I think the overlap was 5%. And so if the overlap is 5%, that means 95% were not replicated in different informatics programs. And so obviously this is an evolving field. We want to now do a new kind of analysis, but the bioinformatic tools don't exist yet. The papers say we're building it, and we're looking forward to it being built, and we hope it'll be available when it's built, even though the community is very good for sharing, I think. But this whole thing brought me to worry about data quality. So I'm a clinician, and when I look at the clinical information of my own cohorts, it's really not very good. And there's all kinds of errors. I don't know how they get introduced there, but I could just imagine huge cohorts of thousands of cases from hundreds of different sites. And I'm quite sure that the quality of the information, at least the clinical information, is a huge problem, at least in my field, which is psychiatry and neurology, as opposed to perhaps in cardiovascular medicine or others where you can actually get accurate measurements as opposed to questionnaires. So I worry a lot about the quality of clinical information. Also, I'm a molecular biologist. Really, that's what I really am in my research world. And so I always worry about using bioinformatic predictions that you assume are close to the truth or reality, and very often there's no validation of whether or not what you're being told you're seeing. I'm not talking about basic sequence data, but things like CNVs, things like many different things that you can find. How close is it to reality? And I've found that whenever we try to do the validation, the biological validation, we always get nasty surprises that what we think we have actually is not correct. So I worry about quality of data, quality of the clinical information. So I guess that was depressing, wasn't it? So nonetheless, I love what I do, and I think that there's huge promise in the field in combining big data, large data sets. I think though that we need to pay more attention to data quality. And especially the clinical quality. And I'll just put in a plug for open access. It was mentioned that it's a good philosophy. I think that open access is got to be what we all strive to do. There are the challenges of data access and basically being able to deal with different data sets. I don't deal with those challenges, but I definitely can appreciate that they are problematic.
Thanks. Before I open it up to the audience for questions, I'm just going to ask if any of the panelists has anything that they'd like to add.
Yeah, I'd like to add one thing. This is actually a very interesting space with regard to science funding in Ottawa, because it actually sits at the intersection of almost all the major funding agencies. So CIHR has it has interests here. NSERC obviously has interests here. Even SHRC to a certain degree has interests here. CFI, Canadian Foundation for Innovation, funds infrastructure here. Genome Canada and the provincial genomes have interests here. And then you have Compute Canada and Canary that are federal, that are also, they have a status similar to Genome Canada. They're federal, whatever, foundations. And so it's really an area where all these different funding organizations that don't always cooperate so well actually can cooperate and can benefit from cooperating. And so CIHR and NSERC have worked together to fund several initiatives in this area. There was one larger one that actually went for handling data, cancer-related data, that was headed up by NSERC but had a CIHR component and a CFI component. So it is an area where all these different federal agencies have a common interest, and it can actually be a good test bed for cooperation between these agencies. That means that it might also be a particularly opportune place to get the attention of government, because government would like all these different agencies to work together better. And so there have been discussions, and these are almost finished, going on for about the past year. It actually came out of a workshop that was held about a year ago in Toronto, I think, bringing together a lot of stakeholders in this area. It was organized by Genome Canada, but it had folks from all these different organizations to really sit down and try to agree on some kind of national strategy that could then hopefully be used to bring more resources in to make sure that GE gets enough storage space on Compute Canada and solve those kinds of problems.
Yep. I have a comment. We heard about data access, and that certainly is an important thing, especially for databases that were created with taxpayers' money. Making those databases accessible to the public is a matter of responsibility and obligation. But I'd like to talk about collaboration, because data could be made accessible, but without collaboration, those data may not come to any good. And I'll give you an example from our own work in Framingham. We were one of the earliest studies that was granted the opportunity for genome-wide genotyping for genome-wide association studies. Roughly 10 years ago, under an institute director at National Heart, Lung, and Blood, who was a visionary, we were given the opportunity to be one of the world's first population studies to get genome-wide genotyping, and we invested in infrastructure. People volunteered their time, even if they weren't getting paid, to develop a pipeline for analysis of multiple traits. And as this moved forward, we thought we would be ahead of the rest of the world, identify genes associated with every imaginable cardiovascular phenotype, and when we were done with the cardiovascular phenotypes, we would then move on to other phenotypes as well. It sounds kind of Napoleonic. What we learned very quickly as we completed the genotyping and started analyzing the data, as we created 19 different working groups to deal with 19 different sets of phenotype domains, we went from group to group, and each group, with only a few exceptions, had not a single genome-wide significant result. And we learned a great deal from that, and journals who are now starting to publish genome-wide association studies were a second motivating force, because they started to refuse to publish genome-wide association results that did not include replication. So cohorts began to come together. My colleagues in Framingham helped found what was called the CHARGE Consortium. It was a cohorts for epidemiology research using genome-wide association data. And as we got our sample sizes up, Framingham alone was 8,000, but it wasn't until we got sample sizes of 30,000 through this collaborative effort, that all of a sudden genome-wide significant results began to accrue. And then through additional growth and collaboration, those consortium sizes went up from 30,000 to 60,000 to 100,000. Some of you saw the recent paper on genome-wide association results for height that involved a total sample size of over 250,000 people to identify enough genetic variants to explain a majority of the missing heritability for a single phenotype. So if it were not for the willingness of these cohorts to collaborate, if it were not for the journals mandating replication, these cohorts would never have come together to accomplish what they could through collaboration, but could never do individually.
Thank you. I'd like to open it up now to the audience. Does anyone have any questions? If you do, please step up to the microphone.
Gil started to make reference to some of the problems of cross-site heterogeneity and compatibility. There's also the issue of cross-time compatibility with the march of technology. Does the committee have any perspectives on this conundrum? You want to maintain as much as possible methodological consistency, but technology is changing under your feet the entire time. So do you stick to outdated technology to maintain methodological consistency, or do you just keep on changing your technology and hope for the best to sort it out down the road statistically? I don't have a solution to this problem.
I'll start on that one and then others can pipe up. So I think I've got two slightly different answers to that. One is, there's actually, if you think even in the genomics world, there's sort of precedent for this with SNP genotyping arrays. 10 years ago, some of the earliest GWAS were carried out using sort of 500,000 so markers. These days, people would tend to use things, about 1 million or maybe 2.5 million markers, and there'll be some people doing whole exome and whole genome as well. What's happened there is that those earlier data sets haven't been thrown away. They are still being used, they're being used very effectively, and that's partly because people have found some ingenious ways of harmonizing the information from those different studies. And the height work that we just heard about, that's not all one platform, that's hundreds of, probably tens at least of different platforms successfully integrated. So it's feasible to do that. The second thing, which is people often sort of worry about holding on to data, it's very precious stuff. One of the sort of the big data mottos is, if you like, is data's cheap, typically, and what might be expensive for you to generate today in 2 years' time will probably, through Moore's law, be a tenth of the price or something like that. We haven't seen the Moore's law in sequencing carry on at quite the rate that we would like to have done to make, say, SNP genotyping completely obsolete. But I think I'm certainly strongly believing that there will be value in going back to those older samples and revisiting them until we get the perfect genomes of all of those samples.
Would anyone else like to weigh on that?
Maybe I just, you got to move with the technology. You can't not do that. So, but often you produce, with the older methods, you produce data sets that you can reduce down to something which is compatible with what you're doing now, but maybe less complete. So I think that using old data is very useful, and you got to keep moving with the technology. And maybe after 10 years or 20 years, the old stuff won't be very useful anymore because there are going to be too many holes, but we do that as well.
I've got a couple questions, so I'll circle around after. So my first question is, I think, for Nancy. Do you feel like there's a bit of a trade-off or a balance? I mean, we've been hearing about data access, and I might be getting it confused with like free access, and the importance of having that kind of accessibility 1,000 genomes is perhaps an example of that. Is there a balance, though, that we have to make in order to maintain, say, programs like yours, where to be sustainable over time, to be able to, like, I wouldn't say charge for access or charge for, there's a fee, but is there a model that we can be considering here and that you guys have implemented?
And the model, you mean in terms of the financial sustainability, or? It is an ongoing challenge. Our general model is that it's centralized funding from various sources, government grants, to cover the base operational costs, and then we cost recover at the margin. I don't see how an infrastructure like this will ever fully be cost recoverable as long as it's accessible, like the user base is academic researchers only. So I think that's another conversation that's ongoing in this space around data access, about who is the user base. As long as it's academic researchers only, it's a very small group, but there's quite a, at the other end of the spectrum is commercial access, but there's a lot in between. So there's ideas about seeing that as an engine of innovation, of economic growth even. Does that answer your question? But I think, when you talk about the longevity of it or the maintainability of it, in a way, almost more important is thinking about the social trust, the public perception, the public kind of social acceptability of it. And that's an ongoing important challenge. And I think in Canada we haven't done a great job at social engagement or public engagement. There's maybe representation on a committee or that sort of thing, but nothing that goes a little bit further. And I think there's a real opportunity in a way of having social acceptability, social enthusiasm drive a little bit more the usage question, where there's the sort of driving the risk management debate. Because I think any surveys that I've seen is that in general the public is quite in favor of using these data, and often the reaction is they're not being used. So that's another layer there.
I think you actually your question touches on a larger issue that funders have to get their heads around, and that's that as science is becoming more collaborative and more reliant on data, databases, common shared facilities and infrastructures, we have to think about a better way of funding those things. I mean, those are often funded from one particular source on an ad hoc basis and for some historical reason, and yet many of these are international resources that are vital to research. And I would love to see somehow some kind of robust international mechanism come together to just sustain these things in the long term. But that's not going to happen tomorrow.
Or even a national mechanism?
Or even a national mechanism.
Or even provincial.
But for instance, GenBank is funded just somewhere in the States, or Ensemble is funded at the Sanger Center. So you have one where it happens to be cited, that organization takes 100% of the cost for it. Yet these are internationally used facilities that really serve the world. And they're probably the way they're funded probably needs to reflect that.
Okay. So my question is, there are a lot of cohorts that were presented here. We know there's many more that exist that weren't even mentioned at all. But it seems like many of these, have a diversity of types of clinical phenotypes, endophenotypes, molecular phenotypes. And so you're seeing really like these pockets and collections of data and cohorts which actually have a lot of different characteristic to them. One of the things that I'm finding is a bit of a challenge is actually trying to weave your way through all of these international cohorts and saying, who has this particular trait? Who has this trait with this molecular phenotype? When can we start bridging this type of information together? So I think on one hand, it's great that we can think about how we might build systems which share, genomic information and, allelic information. But I think, at a higher level, it'd just be nice to know what phenotypes people are actually measuring.
In some areas, there are maybe better databases for that kind of, like a registry of cohorts. So in Canada, actually, Shu Lee really spearheaded this. We have found a small amount of money within CIHR to start to fund setting up a registry of at least Canadian cohorts, so that who's got what and how you access it might become a little bit less of a challenge in a year or two.
I'd love to see that under the same way as like the Global Alliance for Genomic Health. We have that kind of infrastructure to explore cohorts in the same way. And so just throw that out there.
Just a brief comment. One element that is being generated within the Global Alliance is a functionality to be able to say who's got this variant. And that would go out to any cohort that's sort of serving their data through it.
Right.
Now that's a very, very specific version.
You can say who has this phenotype. That would be,
yes.
Yeah.
So who has this phenotype is a much more exciting, more challenging thing to implement, but definitely should be done. I agree.
There has been recent agreement on a common language for human phenotypes.
Yeah.
There's like 2,300, a controlled vocabulary that can be used computationally. So that is the first step, and this is partly through Global Alliance. That is the first step, I think, toward building what you're asking for.
Gil, yeah, indeed. You mentioned...
Do we allow repeats?
Yes,
just
we
keep
do.
circling around, and maybe Stephen wants to come back. You mentioned that, at the beginning of this session, this anecdote in the hospital setting where with an unknown phenotype or unknown disease, you went forward and did the genome sequencing, the exome sequencing or whatever. And still, at the end of the day, it still had to be a group discussion about how to move forward with the clinicians and the bioinformaticians and so on. Does that speak to potentially a failure right now, or a lack of tools or adequate tools, either from the geneticists or the statisticians? Maybe we're not using the right parametric or non-parametric approaches. Have we oversold something? I can keep going, but yeah. Okay.
Yes, it's clear that we've oversold big data and we should just not do it. No, so it's actually, so I'm going to argue it's the opposite, it's a lack of data. So the variant, we all know that if you sequence an individual, you'll find loads of variants that look potentially interesting. So if you come in with a particular presentation, you'll find 10 different stories that you might want to follow up. And that's only looking at the coding part of the genome. The regulatory part is another matter. So the only way you're going to get better at sifting through that set is to have more understanding of what variants do to phenotype and a better understanding of what variants there are in that population, sort of broadly matched for ethnicity, for example. That will only come through having more and more data. The reason that, and that will help us a long way, that will help us get a long way, but it's certainly not going to be the whole thing, because I'm not a clinician, but I've sort of been around clinical geneticists long enough to understand how much heterogeneity there is both at the phenotypic level. The same mutation or mutations in the same gene can have a huge variety of phenotypes. There's heterogeneity at the genetic level. The same phenotype can be caused by mutations in many different genes. Then there's heterogeneity in how information is recorded, and that is a major challenge, particularly in rare disease, where you may be needing to ring up your friends in Montreal to try and find other individuals with the same presentation. So big data hasn't been oversold, but I think maybe there's an unrealistic expectation about how quickly you can turn what is a very powerful platform for discovery into a powerful platform for clinical diagnostic and intervention. I think we heard some lovely examples. Tom Hudson was talking about some cancer work earlier. That is the sort of thing where there really is something that could be put into clinical practice today, and that's very exciting. Realistically, genomics isn't largely at that point, although it's making inroads. So, for example, in the study I was talking about, These are all individuals for whom standard clinical genetic testing had failed. We got a success story in about 30% of them. And then in another 30%, we've got something that is being followed up. And it's probably true and it's useful information, but it's going to be another few years and a lot more data before we have a definitive answer.
So, yeah, I think I would agree. This is a temporary situation. We're at the beginning. We had the Department of Genetics had a retreat and identified the evil VUS. And VUS is variant of unknown significance as the fastest growing reason for consultation in genetics. And a VUS is basically a variant that's been found that we don't know if it's disease causing or not. It might or it might not be, but we don't have the information. So it's a lack of data. And eventually, with better phenotyping, those VUSs will decrease and we'll end up with more certainty, at least for the penetrant rare diseases. For the common diseases, then that I think the jury's, that's going to be more complicated. But certainly for VUSs, it's a problem of data, not enough data.
So more data could mean, well, like Dan was saying, increasing sample size and capturing more effects, but I mean, that's kind of one sort of solution as well. Is it your opinion, for example, do we have the analytical tools? Let's say we were able to increase not just data sample sizes, but the amount of information we have for any one individual or set of individuals. There must be room for more development in that context as well.
Yes.
And I think I would also argue just uses. The more that a given data set is used, the more that's known. It's sort of this iterative snowball effect that there's actually a lot of data that exists today that is just underutilized.
I wonder if the data users on the panel could comment on their experiences regarding finding and recruiting staff to perform the analysis. Of course, if you have big data sets, they're only useful if you can perform the analysis. And that requires a high degree of skill in terms of database management, programming, and bioinformatics. So what are your experiences in terms of recruiting high-quality programmers away from industry? For example, are the bioinformatics training programs doing a good job of training bioinformaticians to adapt to a very rapidly moving technological field?
Should I go first? So it's clear that in this field we're up against Google. I lose people to Google, to financial institutes. I lose them sort of to any place that can offer large salaries to work on problems that involve numerical analysis and computing, of course. What's actually really reassuring and very exciting is that we have people coming the other way too, and no small number. People who are fed up with actually using their analytical and computational skills simply to try and improve the targeting of adverts, or simply to try and improve the margins on some transaction by a fraction of a percent. So it's a two-way street, and I think actually one important point that certainly I'm keen to try and facilitate is to make the interface between commerce and the academic side a little bit thinner, so that people can go from one place to another a bit more easily, and go and come back and go and come back, for example. So you can't compete. I think you have to sort of go with it. Having said that, people like Google and Apple and Amazon have been very, very strong participants, maybe not Apple, but the other two in the Global Alliance, very engaged. And so we actually benefit from that partnership. Having said all of that, it is a complete headache. Recruitment, it's in my experience, is the number one headache. And the training programs are bioinformatic training programs is not what I need. I need people from maths, stats, computer science background brought in and excited about biology and then sort of helped to work on problems that they find exciting and I want to see solved. I don't want people trained in the use of one piece of software that will be obsolete within 6 months. So it's really important to sort of bring those people who are fundamentally quantitative, fundamentally good at programming, right into the heart of this field. What we have is a very, very exciting field, and so attracting those people isn't too hard once you actually expose them to what it is we're trying to do.
So to follow up on the question of the VOS and that if we need more data to elucidate these questions, there's all kinds of data that we could get to try to figure out these questions. We could get bigger cohorts, we could do more model organisms, we could do in vivo mutagenesis and cellular phenotypes. So what kinds of additional data would you guys like to see if we are to answer these questions? Like, where should we put the money in priority to go and answer these questions where there's not yet enough data?
The idea is that if it depends on what it's going to be used for. If it's going to be used for to make decisions on the fate of a person and on a fate of somebody's pregnancy, then we need very high quality data, and so that's kind of the highest bar. But there's everything in between. I mean, there could be large studies that look at variants and look to see if these are functional or not. At least, is it a functional variant or not? That would be a first thing. Does it affect expression? Does it affect something? And that's one kind of data. The other data is better information on the phenotypes. I mean, there's been attempts to link up all the diagnostic labs in the world that are doing exome sequencing, and there's probably been tens of thousands that have been done. And often they have pretty good clinical information. And so if somehow we could standardize all this information, put all this information together, we probably would get closer to having that kind of high quality information we need. But of course, all the other things that you mentioned, it's all good. It's a gradual accumulation of data that leads to greater and greater certainty, and that's all it is.
I have one more comment here. I think if we increase numbers and do more research, we will eventually end up with a certain percentage of the population that has genetic data somewhere in a scientific database that is mined. But there is much more information in these data than is used for this particular purpose. That's neurological disease or cardiovascular disease or inflammatory bowel disease or whatever. Much more information that might be of use for that particular person that contributed their sample towards the study. So I think we might also start thinking how we can give this information back to the patient, how we can include these massive amounts of data in the electronic medical records so that these data can be used by the clinicians who are working with these patients. And I think we have to develop clinical pipelines, clinical grade pipelines, so we can pull these informations through so that we can make this available for the patient and for the clinicians caring for these patients. And I'm especially thinking of information that was maybe not relevant for the study, but maybe relevant for the patient.
Hi, I just wanted to follow up on the point that was just made by Simon Warby, which is an issue that's very close to my heart. How does one maintain high quality bioinformatics, big data analytics people? And I think it's not just the pressure from Facebook and Google and who's stealing our best people. It's the university systems and the funding agencies don't have mechanisms to find a niche for those people. They don't fit traditional departmental criteria. we need to be more agile in how we find a career path for these kinds of people in the university system with funding programs for them. So I think one of the things that we could do as a community is develop white papers and put some pressure on the traditional academic authorities to build a program for these kinds of people. Otherwise, we're going to end up with all the people who can't get jobs in industry. We're going to get the second bananas. That's not what we want.
I'd like to make a comment about new approaches that might reduce the overall computational burden for analyzing big data. I mentioned that we're part of a number of consortia that unite their data for genome-wide association or whole exome or whole genome sequence data, transcriptomic data analysis, etc. Because of consent provisions, it had been necessary when we have a consortium of 17 studies for each of those 17 studies to do a complete analysis of whole genome data or whole exome data, and then find one or two analysts who could meta-analyze the data because we were unable to transport individual study data for analysis because of the limitations of informed consent provisions. We're now working with a new approach that would allow individual studies to upload their data to a secure site on the cloud, and someone called it a black box or a glove box method, where as each of those same 17 cohorts upload their data, a set of tools exist for analyzing those data as if they were individual participant level data now being analyzed, and now one analyst can analyze all 17 cohorts instead of having 17 cohorts, each of which has to train an analyst to analyze whole genome sequence data. So these new approaches of creating data portals and centralized cloud-based computing where the person doing the analysis doesn't actually have access to individual participant level data from each of those 17 cohorts, but can press a button to allow all of those analyses to take place. And that literally can reduce by 17-fold the number of people who have to conduct the analysis for a single consortium effort, and reduces the amount of time required for that to happen quite dramatically.
So I think we have time for one more question.
Thank you very much. I have a different question. I'm Anne-Monique Nu from Saint-Justine. Following on your point about bringing back the data of all these magnificent cohorts to the patients, as well as the constraint of the regulations we have for sharing data or publicizing some information and things like that, the worry there are the regulations that we haven't made but are there. How do you see, and have you started to involve the participants of the cohorts into designing the relevance of the questions and the relevance of the regulation too? So more precisely, for example, in we're in a context where we're being pushed toward involving patients into relevance of questions, research questions, relevance of writing the study design, and also we know, for example, patients have been very instrumental into advocating for certain research, for example, to go on or to be funded. So, for example, in Framingham, I know there's a sign at the entrance of the town, home of the Framingham study, if I'm right. So this means they take care of that. They worry about the continuation of this study. Has there been a positive element that's been integrated into pursuing these cohorts?
Yeah, that really is an important question. I'll try to respond a little bit, and that's the realization that our study could go on if any of our senior investigators retired or was attracted to go to Google or Apple, we'd be able to continue just fine replacing any investigator in our study. What we can't replace is our study participants. And we have worked tirelessly to try to maintain their loyalty, to try to always remind them of the importance of their contribution. We have a regular newsletter that we mail out to study participants, as well as now maintaining a website in which we inform them about important scientific information coming out of the data that they are contributing, publications, and in lay terms, what these publications contained and how this helped advance scientific knowledge. You know, a reporter once asked me, tell me, why is it your study participants keep coming back time after time to be poked and prodded in your exams? I went through a very long-winded answer, and she cut me off and she said, I think what you're really trying to say is that they're motivated by altruism. And I said, gee, I wish I had said that. She said, I'll quote you anyways, having said it, that people really would love, everyone would love to be, you know, Mother Teresa if they could. They don't have the time to devote their entire life's energy to being able to help humanity. But by contributing a small amount of time every few years to be poked and prodded a few times, if people understood that that little effort allowed them to advance the well-being of the people around the world, they're willing to make that sacrifice. And just simply reminding people over and over again that the contribution that they're making makes a huge difference to many people really does stir up that enormous sense of loyalty and sense of altruism.
In fact, my question was a little beyond that, is have you asked your participants to help you advocating for funding more precisely? Do you have a committee of participants that are going after whoever, politics or whatever, to say, we as patients also say this is important, or I shouldn't say patients, just citizens?
Yeah, so we do have a participant organization called the Friends of the Framingham Heart Study that solicits small donations that come back that allow us to send fellows to national meetings when we don't have the resources ourselves to allow that to happen. We have participants who send letters on our behalf, and when funding was very difficult a year and a half ago, the senator from Massachusetts, Elizabeth Warren, stood up on the floor of the Senate to complain about funding cuts to the Framingham Heart Study. And so all those little things perhaps make a little bit of difference by mustering up enthusiasm for maintaining funding. And you know, the reality is that funding is difficult, and resorting to every possible effort perhaps does make a difference in the end.
So with that, I'm going to wrap up our session, and I want to say thank you to everyone for staying till the bitter end, and thank you to our panelists for having thoughtful things to say at the bitter end. So.